Good day, grade 12 learners, and welcome to this video where we will be focusing on the process of DNA replication as well as DNA profiling. In the previous video, based on your exam guidelines, we touched on the following: the location of DNA, the brief history and discovery of the DNA molecule, the structure of DNA, three components of a DNA molecule, a stick diagram of a DNA molecule to illustrate its structure, and lastly, we looked at the functions of DNA. In this video, the focus will be on DNA replication as well as DNA profiling. In DNA replication, based on your exam guidelines, we will touch on the process of DNA replication. We will look at when in the cell cycle it takes place, where in the cell cycle it takes place, how DNA replication takes place. Remember that the names of the enzymes are not required. You do not need to know the names of the enzymes that's involved in DNA replication. And then we'll look at the significance of DNA replication. Secondly, we'll look at DNA profiling, the interpretation of DNA profiles, and the uses of DNA profiles. So we'll first start off by looking at the process of DNA replication. We'll speak about when in the cell cycle it takes place, where it takes place, how DNA replication takes place, and the significance of DNA replication. So firstly, what is DNA replication? What do we mean when we're speaking about DNA replication? So DNA replication is the process during which a DNA molecule makes an exact copy of itself. So that DNA molecule that we learned about in the previous video, the DNA molecule that is a double-stranded molecule that comes in the shape of a double helix, we're simply saying that DNA replication is the process during which that molecule is going to make an exact copy of itself. What is the significance of DNA replication? So why is it important that DNA replication actually takes place? Why does DNA need to make an exact copy of itself? So number one, it ensures that each daughter cell gets an identical copy of the DNA molecule after mitosis. So there are two basic reasons as to why DNA replication is important, and the first one, it ensures that each daughter cell gets an identical copy of the DNA molecule after mitosis. Now just a quick revision on the process of mitosis that you touched on in grade 10. You would have remembered that in grade 10 when you spoke about mitosis, there was different stages of mitosis. You had interphase, prophase, metaphase, anaphase, and telophase. Now, when we are looking at DNA replication will occur inside interphase of the cell cycle. And the first reason as to why DNA replication is important, it ensures that each daughter cell gets an identical copy of the DNA molecule after mitosis. What does that mean? In interphase, DNA is going to undergo the process of replication. So that means that each individual chromatid that you initially have here in interphase, it's going to make an identical copy of itself. So you have one pink and you have one blue. So in the next phase, you will have two pinks and two blues. Why? Because DNA underwent the process of DNA replication. So if you move from number 1 to number 2, you will notice there are now two pinks. You have two chromatids joined by a centromere to form that chromosome. And the blue as well, you now have two blue chromatids joined by a centromere to form that chromosome. And how did they have two chromatids? They underwent the process of DNA replication. So at the end of telophase, each daughter cell is going to have an identical copy of the DNA molecule after mitosis has taken place. That is the explanation of that first reason, the significance of DNA replication. The second significance, it ensures that each daughter cell gets the correct number of chromosomes after mitosis. So what does this mean? At the end of mitosis, after the last phase, telophase, each daughter cell will have the correct number of chromosomes after mitosis. And generally in humans are going to have 46 chromosomes. So at the end of mitosis, each daughter cell is going to have that 46 chromosomes. And why does it have 46 chromosomes? Because the process of DNA replication ensured that the correct number of chromosomes was found inside the daughter cells. So those are two reasons as to why DNA replication is important. You need to know these two reasons. It can be tested and it is examinable. Right, now when in the cell cycle does DNA replication take place? So you would have remembered previously earlier on, we mentioned that DNA replication took place in interphase of the cell cycle that occurred before prophase. Where in the cell cycle does DNA replication take place? It takes place inside the nucleus of the cell. Now, why are we saying that it takes place inside the nucleus of the cell? Now, remember in the previous video, we said that there are different locations of DNA. DNA is found inside the nucleus, the mitochondria. It's also found inside the chloroplast of plant cells. Now, the reason as to why DNA replication occurs inside the nucleus is because majority of our DNA is found inside the nucleus. So that is the reason as to why DNA replication occurs inside the nucleus. But all that you need to know is, you need to know that DNA replication occurs inside interphase of a cell cycle and DNA replication occurs in the nucleus of the cell. We will now move on to describing the process of DNA replication. So I'll first go through the 5 steps of DNA replication and thereafter I will use a diagram to assist the explanation. So firstly, number 1, the DNA double helix is going to unwind and unzip. The weak hydrogen bonds between the nitrogenous bases are going to break and 2 DNA strands are going to form, 2 separate strands. Number 3, each original DNA strand will serve as a template on which its complement is built. Number 4, free nucleotides build a DNA strand onto each of the original 2 DNA strands by attaching to their complementary nitrogenous bases. Adenine will join with thymine and guanine will join with cytosine. Number 5, this results in 2 identical DNA molecules. Each molecule consists of 1 original strand and 1 new strand. So let's have a look at the diagram to assist us in understanding this process. So step number 1, if we look on top, we will notice that we have the normal DNA double helix structure. That DNA double helix is going to unwind and unzip. The weak hydrogen bonds that are found between these nitrogenous bases are going to break. You are going to have 2 separate strands forming. Both strands will serve as a template strand. So you have 2 separate strands forming and both strands will serve as a template strand. Then you are going to have your free floating nitrogenous bases that are going to come from the nucleoplasm and they're going to align themselves complementary to their base pair. I'm going to repeat that. You're going to have free floating nitrogenous bases coming from the nucleoplasm and they're going to align themselves complementary to their base pair. So adenine will align next to thymine and guanine aligns next to cytosine. The weak hydrogen bonds will now form between that complementary base pairs on either side. The weak hydrogen bond will now form. The DNA molecules are now going to coil up again until they form their double helix structure. And at the end, you have 2 identical DNA molecules. So you have 2 identical DNA molecules and the one is the exact copy of the other one. So originally you only had one DNA molecule at the start, but at the end of DNA replication, you have two identical DNA molecules. This is a process that will definitely be tested in your final exams, in your March control test. So please ensure that you are able to describe the process of DNA replication. This could possibly be a 5 to 6 mark question in your March control test, or even in your June exam and your final exam as well. If you have a look at the following video, it can simply assist you in understanding the process. So I'm just going to play it and then I'll explain to you. So if you have a look on the left hand side here, you will notice that the DNA double helix is unwinding, and the weak hydrogen bonds have broken, and you have two separate strands forming. You have free floating nucleotides, nitrogenous bases coming from the nucleoplasm, and they're going to align themselves to the complementary base pair. So you see how they are all starting to pair themselves there until they have formed another DNA strand. So that is basically what happens in the process of DNA replication. Now, sometimes you will notice that mistakes are going to happen in the process of DNA replication. So therefore, we're going to move on now to the copying of DNA and mutations. Now, sometimes mistakes are made in the copying of DNA and the sequences of bases is changed. So sometimes you will notice that when your nucleotides are going to come from the nucleoplasm, they may perform some mistakes, and these mistakes are referred to as a mutation. So when you change the sequence of nitrogenous bases, you're going to cause a mutation, and this mutation can actually have a negative effect on the process of protein synthesis because it can cause a different protein to form. So how does this affect the genetic information carried by the DNA? So if the sequence of bases on the DNA molecule is changed by mistake, then the genetic information is also changed. What do we mean by that? Remember in the previous video, we said that one of the main functions of DNA is that it provides hereditary information. And then we also said that DNA provides the code for protein synthesis. So therefore, it follows that if you are going to have a mistake occurring and you are going to have a mutation, then the DNA code will be a different DNA code. And if you have a different DNA code that's been provided for the process of protein synthesis, then you are going to have a different protein that could possibly be formed. And this type of change is going to be referred to as a mutation. So based on the exam guidelines, we've touched on the process of DNA replication so far. We looked at when in the cell cycle takes place, where in the cell cycle takes place, how DNA replication takes place. Remember, this is a possible 5 to 6 mark question where you will need to describe the process of DNA replication. Please make sure you are able to do that because that is easy marks that you can be awarded for in a control test or in an exam. Lastly, we also looked at the significance of DNA replication, and there were two reasons as to why DNA replication is important. We will now move on to DNA profiling. In DNA profiling, we'll look at the interpretation of DNA profiles and the uses of DNA profiles. So firstly, what is the definition of a DNA profile? What is a DNA profile? The process by which the DNA of a person is mapped. Or you can say the process by which the DNA sequence of an individual is determined. Now, what do we mean by the DNA sequence? So remember we said that DNA is going to hold hereditary information. So if you are looking at your DNA profile, you're taking all your hereditary information, your DNA characteristics, and you're going to be arranging it in the form of bars. And we'll look at later on, we'll see exactly how this is going to occur. So if you're defining the DNA profile, you can either say the process by which the DNA of a person is mapped, or you could say the process by which the DNA sequence of an individual is determined. So what are some of the reasons as to why we are going to have DNA profile? What are we going to be using it for? So some of the uses of DNA profiles are as follows. Firstly, to investigate crimes or solve disputes. Secondly, to identify organisms from their remains, that is normally dead remains. Thirdly, to identify missing persons. So if you're trying to identify a missing person, you could possibly use their DNA to help to identify them. For paternity testing, so if you are trying to determine who exactly your father is, you don't know exactly who your father is, you could use your DNA and your father's DNA, and then if the DNA are the same, you can come to the conclusion that that is actually your father. Then to identify family relationships other than paternity, things like siblings or cousins. And then lastly, to establish matching tissues for organ transplants. Please ensure that you are able to understand and memorize these reasons, because they could possibly ask you some of the uses of DNA profiles in a test or in an exam. What are some of the disadvantages of using a DNA in a court of law? So let's say you have to go to court and you are trying to use DNA as evidence for proving your case. Then what are some of the disadvantages of using DNA? Now, firstly, human error could cause the DNA evidence to be mixed with another person's DNA. So sometimes when they are analyzing the DNA samples, and they could possibly make mistakes. So therefore we say that human error could cause DNA evidence to be mixed with another person's DNA. And then possibly when you are standing in court, you would present the wrong person's DNA, and that is how a mistake could occur. Secondly, DNA evidence could be planted to frame a suspect. Now, when does this normally happen? Let's say there's going to be a crime scene that occurs. Maybe there was a murder that took place, and somebody decided that they wanted to murder another person, but now they want to frame somebody else for that murder. They could take that person's DNA and plant it on the crime scene, and then they could possibly frame another person who could now be a suspect for that murder. So secondly, DNA evidence could be planted to frame a suspect. Thirdly, the suspect could have an identical twin with an identical DNA profile. Now, what does this mean? Generally, the DNA that you have inside your body, nobody on this earth would have exactly the same DNA as you do. However, if you do have an identical twin, that is the only time that somebody could have the same DNA as you. So if a suspect in a specific case has an identical twin, then it could be a disadvantage if you're going to use their DNA in a court of law, because the identical twin would have the exact same DNA as they have, and then it would be difficult to prove that that particular person is going to either be innocent or guilty in this particular crime that you are trying to solve. So how is a DNA profile made? So if you want to make a DNA profile, a sample of DNA is first extracted from body tissue or from fluid. The sample is broken into smaller pieces. The small segment of DNA is exposed to X-rays. A pattern of black bars are left on the X-ray film, and this is known as the DNA profile. So you don't need to know exactly how a DNA profile is made. This isn't really examinable, but it's just explaining exactly how we go about making a DNA profile. The previous slides that were shown, those things you need to know. You need to know the uses of the DNA, and you also need to know the disadvantages of using DNA in a court of law. This question could possibly pop up as well. Now, when we are taking DNA, when we are trying to extract DNA from our bodies, what are some of the sources? Where can we actually obtain DNA from? So I've just listed four basic things, four basic locations in the body where you could extract DNA from? Firstly, you can extract DNA from hair follicles. So hair follicles coming from your head. Secondly, from a blood sample. So if you look at this injection, you could basically remove blood from your actual body, and when you place it into a little tube, and a blood sample can then be used to analyze your DNA. Thirdly, saliva. You could take saliva from your mouth, and the saliva sample can be used inside your saliva. You can also obtain DNA. And then lastly, from semen. So if you are a male, you could extract the semen from your body, place it inside a little sample, and then the semen also comprises of DNA. So please remember the following. They could possibly ask you where you could obtain your DNA from. It could be from your hair follicles, blood sample, saliva, as well as semen. Now lastly, if we're trying to interpret a DNA profile, how could we possibly interpret a DNA profile? So if we have a look on the left-hand side of this dotted line, you'll notice you have a victim and you have a crime scene. And on the right-hand side of the dotted line, you have suspect 1, suspect 2, and suspect 3. Now, we would normally try to interpret the DNA profile if we are trying to solve a certain type of crime. So let's say we are on a particular crime scene. This is the DNA we found on the crime scene. This is the DNA of the victim on whom the crime was committed. And then on the right hand side, we have 3 possible suspects. Now we are trying to find out which of the 3 suspects is possibly going to be the guilty party. Now if we are trying to figure which suspect is guilty, we need to look at the DNA profile. So this arrangement of bars that we have in each suspect and of the crime scene and of the victim, these bars are the DNA, and this is referred to as a DNA profile. We want to try to match each of these bars with the crime scene, DNA profile of the crime scene, and DNA profile of the victim. If we look at the first bar of suspect 1, we see that it only matches the crime scene, but it does not match the victim. Therefore, we ignore that bar. We go to the second one. You notice that this bar matches the crime scene and the victim, so I can place a tick on that one. If I go to the next bar, it only matches the victim. I can't take that. I go to the next bar, it matches only the crime scene. You therefore cannot take it. If I go to the last bar, you can see the last bar matches the crime scene and the victim. There are therefore 2 bars that match for suspect 1. So at the bottom, I'm going to write that there are 2 bars that matched for suspect 1. I now go on to suspect 2. The first bar matches both the crime scene and the victim, so I can tick it. The second bar only matches the crime scene. I can't take it. The third bar matches the crime scene and the victim. Fourth bar only matches the crime scene. I cannot take it. The next bar only matches the crime scene. I can't take it. The last one matches the crime scene and the victim. Therefore, I tick it. And how many bars were matching? There were 3 bars matching. Now I go to the third suspect and I see that this bar does not match any the crime scene or the victim. Second bar matches the crime scene and the victim. I can tick it. Third bar does not match anything. Fourth bar only matches the crime scene, so I can tick that. Next bar does not match anything, and the last bar only matches the victim. Therefore, I can't tick it. So there were 2 bars matching for suspect 3. Therefore, I can conclude that for this particular crime that I had, suspect 2 was going to be the guilty party, since suspect 2 had the majority of these bars matching with the crime scene and the victim of this particular criminal activity. So this is basically how you would go about answering the question if you need to interpret a DNA profile. So you will need to complete it in exactly the same procedure that I have done now. You would normally use your ruler. You'd place your ruler on the bar and try to see which of the bars are actually going to be matching. You tick it off exactly the way I did, and you place the numbers at the bottom of each suspect, and you will then figure out which suspect is going to be the guilty party. So in summary, based on the exam guidelines, in this video we touched on DNA replication, the process of DNA replication, as well as we looked at DNA profiling, the interpretation of DNA profiles, and the uses of DNA profiles. In the next video, we will be looking at RNA. We will be looking at RNA as a type of nucleic acid. We'll look at the location, structure, and functions of RNA.